Okay, thank you. Today's topic is immunoprophylaxis. I hope you all are fine. Immunoprophylaxis means uh, prophylaxis, the word prophylaxis means prevention of something that is called prophylaxis. Immunoprophylaxis means prevention of a disease by inducing immunity. Immunity means body's own resistance against infections. So it is a great contribution of microbiology of medicine to the humankind, the immunoprophylaxis. And in immunity also, this is one of the most important, most effective, most beneficial, most practical aspect of not only the immune system but also the whole microbiology. So because there are certain diseases which can be completely preventable, which are completely preventable by vaccination, like diphtheria, pertussis, tetanus, measles, mumps, rubella, poliomyelitis. The vaccination against these diseases, the immunization against these diseases is so much effective that in some countries they have virtually eliminated all these diseases, in particularly the developed countries. And some other diseases like influenza, hepatitis B, pneumococcus, and Haemophilus influenza, we have achieved a great benefit for the humankind against these diseases. But there are diseases for which we do not have any effective vaccination yet. Despite we want to make a vaccination against these diseases like malaria, leprosy, helminthic diseases, and HIV. So, despite our best efforts, we still do not have vaccines against these diseases. So, immunization can be done by two agents. One is vaccines, as you know, the other is immunoglobulins. Vaccines and immunoglobulins, these two are the major main immunizing agent. Vaccine induces active immunity, and immunoglobulins means you are providing passive immunity, preformed immunoglobulins to the patient for fighting against the infection. So vaccine is contains antigen, and in a very simple way, vaccine contains antigen in a different preparations, and immunoglobulins contains antibodies. That is in a very simple way to put it. So vaccine will induce active immunity, whereas immunoglobulin is preformed antibodies which are given in certain emergency situations. So these are two immunizing agents. Now coming to the vaccine, the word vaccine has come from vaka, which means cow. I think you all know the story of Edward Jenner, how he successfully immunized people against smallpox with the help of a cowpox virus. So he was the first person to use the effective vaccination. So this term has come from there. Vaccine means something you derived from cow, something derived from cow. Because at that time there were two type of immunization prevalent for smallpox. One was that was invented by Edward Jenner from the cowpox virus, and another was which was prevalent in China and India, that was inoculation of a smallpox virus itself in skin or by inhalation. That was called variolation. So vaccine term has come from there because it is first it was derived from the cowpox virus. Uh, in general, vaccine is a preparation from an infectious agent, and when it is administered to humans, it induce protective immunity. It can be prepared from a live modified organism, inactivated or killed organism, extracted cellular fractions or toxoids, or combination of any of these. And the recent Preparations are subunit vaccine, recombinant vaccine, RNA and DNA vaccine, and we are going to talk about them. Suppose this is a pathogen. In this picture, I am showing you a virus, but it can be any pathogen. It can be a bacteria or virus, RNA virus, DNA virus. So when suppose we have completely inactivated this pathogen, we have almost completely killed this pathogen, and we are administering it as vaccine, then we call it whole inactivated vaccine or a killed vaccine, because the pathogen in this vaccine is killed. It is not going to multiply inside the body, but still it has immunogenic capacity. It can induce immune system, but it cannot cause disease. Another way of immunization is you have attenuated the pathogen. Attenuated means you have taken away the ability to produce a disease. The pathogen is alive, it can divide, but it will not cause disease. So these type of pathogens are called attenuated or modified pathogen or attenuated pathogen, and we call these vaccines as live attenuated vaccines. So these are kill vaccine where the pathogen is completely killed, and these are live vaccines in which the pathogen is alive. It can multiply inside the body, but it will not cause disease. Another is when we take a fraction of the pathogen, important protein of the pathogen, which will antibody against this fraction or this protein will be protective for infection. We call it protein subunit or a subunit vaccine, in which we have taken one part of the pathogen. In this, we have taken the spike protein of this virus, but it can be any other part. It can be a polysaccharide in bacteria. In case of bacteria, it can be a capsular polysaccharide or a cell wall polysaccharide or a surface antigen in case of virus. So this is a portion of the pathogen, a subunit of the pathogen. So this is called subunit vaccine. In some viruses, we remove the genetic material of the virus, and this particle is called virus-like particles, and they are completely immunogenic, but they cannot divide since they do not have any genetic material in them. They can also be used as immunizing agent. Sometimes what we do, we take the genetic material of the pathogenic organism and we use it as immunizing agent. In this case, it is DNA or RNA. We can take the DNA and RNA of the pathogen and inject it into the body of the person, and there this RNA, for example, it will act as mRNA. It will induce protein synthesis for the desired protein, and that protein will elicit immune response. So these are called DNA or RNA vaccines. Another way is we take this genetic material and we inject this genetic material into a non-pathogenic organism. And this non-pathogenic organism will show these antigen on its surface, and it will act as a immunization agent, and this is called viral vectors. Sometimes we also use bacterial vectors, or we call them recombinant vaccine, because we have converted the genetic material of one organism in order to produce a desired protein. So these are called recombinant vaccines or vector vaccines. In case of bacteria, some bacteria they produce toxin, and sometimes it is the toxin itself. It is only the toxin that is responsible for the disease. In those cases, we can use toxin or modified toxin. We call them toxoid as a immunization agent. So we have a killed vaccine, live attenuated vaccine, Subunit vaccine, DNA vaccine, vector vaccine or recombinant vaccine, and toxin or toxoid or virus-like particles. Now coming to the live vaccines, they are prepared from live organisms, and what we do, we culture these organisms again and again uh, in another host. For example, there is a virus that will cause disease in humans. What we do, we take that virus, we culture this virus into some other organism, some animal. And when we repeatedly do it again and again and again, after several passage, this virus or this pathogen, it loses its pathogenicity to humans. It modifies itself in order to survive in that organism, in that animal, and that modified organism is no longer capable of causing disease in humans. So this one is called attenuated, live attenuated organism or live attenuated virus or bacteria. They retain their immunogenicity, but they lose their capacity to cause a full-blown disease. So the advantage of using a live vaccine or a live attenuated vaccine is they are very potent because they will mimic a infection, a natural infection, and generally a single dose is sufficient to provide full immunization. One exception is the polio vaccine. In polio vaccine, the OPV vaccine, we give at least 3 doses, and they produce a durable immunity because they are mimicking the infection, but this duration is not always as long as that of the natural infection. There are certain other consequences of being at a live vaccine. One is when you are administering 2 live vaccines, there should be at least a gap of 3 weeks between them. And if you have to do it on you have to do it simultaneously, do it on separate arms or separate places in the body. And live vaccine should not be administered to immunocompromised patients. Immunocompromised patients means whose not normal body's immunity is down because of some medication, cancer, or some other disease. So in those patients, immunocompromised patients, one should not administer the live vaccine because it is possible that because of the immunocompromised state, this live vaccine will remain inside the human body for a very, very long time, and it can revert back to its natural condition. It can become again an active virus. So it should not be administered to immunocompromised patients. Pregnancy is another contraindication, but we can use live vaccine in pregnant ladies if the risk of getting the disease is much higher than the risk of the live vaccine. Examples of live vaccines: BCG, measles, oral polio, anthrax vaccine, cholera vaccine, plague vaccine, salmonella vaccines. There are live vaccines available against all these diseases. These are some of the examples. And against, for example, cholera and salmonella, there are vaccines which are kill vaccines also, or subunit vaccines also. But I'm here using them as an example of live vaccine. Live vaccine. Now coming to the killed vaccine. Killed vaccines are those where we have completely inactivated the pathogen with the help of with the heat or chemicals, because when you heat the proteins of the pathogen will coagulate, will denature, or some chemicals like formaldehyde, we can or alcohol, we can denature the protein and of the organism, and we can use it as completely inactivated or killed vaccine. They stimulate again active immunity. They are safe because there is no chance of infection in killed vaccine, and they are less efficacious than the live vaccines because they are killed. They will not induce any infection. They will not they will not mimic an infection. They will not multiply inside the body. So they are less efficacious than the live vaccines. and because they are killed, they will require multiple doses, at least 2 or 3 doses. And after some time, you also need a booster dose. Booster dose is also required, and the duration of immunity lasts from months to years. So these are kill vaccine. They are very safe. They induce active immunity, and you require multiple doses, 2 to 3 doses, and they are less efficacious than the live vaccines. Another type of vaccines are toxoids. Toxoids. They are particularly used for the bacterial infections, and even in bacterial infections, they are used in those bacterial infections where the pathogenicity of the organism is because of its toxin, mainly because of its toxin. Because if you use them against those organisms which are invasive, these vaccines will not work against those organisms. As you know, some bacteria are invasive, some bacteria are toxigenic, some have both mechanism of pathogenicity. So they are very useful against those organisms where the pathogenicity is only because of the toxin. Only because of the toxin. So such two bacteria are diphtheria and tetanus bacilli. In diphtheria and tetanus, the pathogenicity is because of the toxin itself. They are not uh, invasive organisms. So what we do, we take the toxin, we deactivate this toxin, and we use it. We call it toxoid. When we modify the toxin in order to make it safe, that it will not cause the disease, so we call it toxoid, and we use this toxoid as vaccination for vaccination, and the antibodies will formed against this toxoid will be active against this toxin also. It will neutralize the toxin and it will prevent the patient from the disease. And they are uh, very effective and safe immunizing agent because it is just a toxoid. It will not induce a disease, so they are safe and very effective. And the examples are diphtheria and tetanus. Diphtheria and tetanus. Another type of vaccine is cellular fraction. It is prepared from the extracted cellular fractions of the pathogen. Examples are pneumococcus vaccine, in which we take the cell wall polysaccharide. Meningococcal vaccine, in which we take the cell wall polysaccharide. Pneumococcus vaccine, in which we take the capsular polysaccharide. And hepatitis B vaccine, in which we take the surface antigen of hepatitis B. They are again very effective and very safe. These are called cellular fraction vaccine. Some vaccines are mixed or combined vaccines, in which there are more than one kind of immunizing agent. For example is DPT. In DPT, there is diphtheria toxin, acellular bacteria pertussis, and tetanus toxoid. This DPT vaccine contains toxoid of diphtheria and tetanus and bacteria pertussis bacteria. So this is a combination of 3 organisms. This is called DPT vaccine. Sometimes we give a combination of 2 toxoid, diphtheria and tetanus, that is called DT vaccine. Sometimes we give combination of diphtheria and pertussis and typhoid. MMR is a combined vaccine, in which we give live organism measles. It is a live attenuated vaccine for measles, mumps, and rubella. And now are available also Pentavalic vaccine, which contains DPT plus Hib plus hepatitis B, HBsAg. It contains 5 antigens in it. These are called pentavalent vaccines. Another type of vaccine is recombinant vector vaccine. Recombinant vector vaccine is where we isolate the antigen coding gene, we insert it into a non-virulent virus or bacteria, and then this non-virulent virus or bacteria will produce the protein on its surface, and it will induce the immune system against the virulent pathogen. Examples of these vectors are adenovirus, vaccinia virus, canary pox virus, and attenuated strain of salmonella and mycobacterium. They can be used as vector. And currently, two vaccines for COVID-19 are also they are recombinant vector vaccine. One is the Oxford AstraZeneca. The trade name in India is Covishield, and in some countries it is also known as Vaxzevria. And in Johnson and Johnson COVID-19 vaccine. Both of these vaccines are example of recombinant vector vaccine. In this Oxford AstraZeneca, the vector is chimpanzee adenovirus. In Johnson and Johnson, it is human adenovirus, and they are available in the market. RNA vaccines. In RNA vaccines, what we do, we introduce RNA into the tissue, and this RNA act as messenger RNA, and it build foreign proteins, the proteins of the pathogen, and it stimulates adaptive immune response. Because RNA is highly degradable inside the tissue, so what we do, we take a delivery system, we take some lipid nanoparticles, and inside those nanoparticles is the RNA, which is delivered into the tissue, and this RNA is taken up by the immune cells, and within the immune cells, this RNA will produce a protein, and those protein will be expressed on the surface of immune cells as antigen presenting cells, and they will induce immunity. The advantage of using RNA vaccine is it takes very less time to manufacture them, and they are highly efficacious, and there are less chance of allergic reaction because we are not using any protein in it. So proteins are usually more allergic. So because we are not using any protein, there is less chance of allergic reaction. Still, because of some people are also allergic to lipid used in the vaccine. So very rarely we can see allergic reaction in RNA vaccines also, but usually they are less. There is less chances of allergic reactions, and because they are non-infectious, this RNA, once it has produced the protein, it will degrade inside the cell, so there is no chance of infection. They are very safe. They are also used for some non-infectious diseases for as therapeutic use. Under trial, they are under trial for the use of therapeutic use for cancer. There are two types of RNA vaccines. One is non-replicating mRNA vaccine and in vivo self-replicating mRNA vaccine. Non-replicating RNA vaccine is very simple. It contains only the mRNA. Once it has made the protein, it will degrade itself. In this self-replicating mRNA vaccine, we also give the code for replication to the RNA, so it replicates itself inside the cell. Advantage one, as I said, it is very safe because it is non-infectious, and one advantage is, which is not there with the DNA vaccine, that it will not integrate itself to the host genome. It will not cause any change to the host genome because it is RNA, and it will degrade once the protein is made. It is efficacy is good. It is well tolerated by healthy individuals. Production is rapid, and they can very easily standardized, and they can be used in order to responsiveness to the emerging outbreaks. For example, first time the RNA vaccines are used for as a COVID vaccine. This is the first time that we have ever used RNA vaccine in history for COVID, and we are using them successfully. So US Mrna and Pfizer Biotech, these two vaccines are in the market. They are RNA vaccines. Important challenge is, one is the delivery system. We need some other delivery system, lipid particles, nanoparticles in order to deliver RNA. And one is cold chain maintenance, because RNA is very easily degradable. We have to use the cold chain for the transport and storage of the RNA virus. DNA vaccines are also similar to RNA vaccine. We inject DNA, it will first uh, convert into RNA, mRNA, and then it will produce the protein. But these vaccines are still under trial. They are not available in the market, and they are still under way with several different DNA vaccines. For example, malaria, HIV, influenza, hepatitis B, herpes virus, and different cancers, lymphoma, prostate, and colon. Another type of vaccine is conjugate vaccine. Conjugate vaccine means in which we combine two antigens. One is a weak antigen, which is the desired antigen, with a strong antigen. So with the help of this strong antigen, this desired weak antigen will also elicit immune response. One example is pneumococcal conjugate vaccine, in which we take capsule polysaccharides of various strains like 1, 3, 4, 5, 6, 7 strains, or some vaccines are giving 23 strains, and we combine these polysaccharide antigens with a strong antigen, for example, a non-toxic recombinant variant of diphtheria toxin. So what we do, we take pneumococcal polysaccharides, we combine it with diphtheria toxin, which is a non-toxic variant, recombinant variant, which is called CRM197, and that is used as a conjugate vaccine. It is very effective vaccines against pneumococcal infection. So these were a different type of vaccines. So we discussed live vaccines, killed vaccine, live vaccine, killed vaccines, and subunit vaccine, and RNA DNA vaccine, conjugate vaccine, and viral vector vaccines. Now coming to the immunization. Immunization is of three types: active immunization, passive immunization, and combined passive and active immunization. Active immunization means we will give antigen that will elicit immune response. Passive immunization means we are giving pre-formed antibodies, or sometimes we also do give cells. We can also give cells. And combined passive and active immunization, in which we give both active and passive immunization. Active immunization can be of two types. One is routine immunization, or immunization of the individuals or selected groups. Routine immunization is part of basic healthcare, which means it has to be given to every individual, every single individual, irrespective of his risk, or whether he belongs to the high risk group or not. It has to be given to every single person. For some vaccines which are given to every single individual, they are polio, tetanus, diphtheria, and measles. And another immunization is immunization of the individuals who are a selected group. For example, high risk group, or people who live in a particular geographical area where certain diseases are endemic, for example yellow fever, or to the people who are traveling to those areas. So these are the immunization of some selected group, and this is routine immunization. Routine immunization, in India, there is a schedule which is called National Immunization Schedule for the routine immunization of all individuals, and one another immunization schedule that is given by WHO, that is called Expanded Program on Immunization, which is followed worldwide by WHO. This is our Indian National Immunization Schedule. This is WHO immunization schedule for the whole world, Expanded Program on Immunization, or EPI. Indian National Immunization Schedule, it is in this, what we do, we give at birth BCG and OPV at birth, and at 6 weeks we give BCG if not given at birth, and we give first dose of DPT and OPV, and we repeat this DPT OPV at 10th week and at 14th week, and at 9 months we give measles vaccine, and after that at 16 to 24 months we give again another dose of DPT and OPV, and at 5 to 6 years, we give a booster dose of DT, and at 10 to 16 years, we give tetanus toxoid. This is, and for pregnant ladies, those who are in early pregnancy, we give the first dose of TT, and if they are already immunized, we give them a booster dose, and after 1 month, we give another dose of tetanus toxoid. We give 2 doses of tetanus toxoid for pregnant ladies. This is the Indian National Immunization Schedule, and in some states, for example Delhi, hepatitis B vaccination is also given as routine immunization, which is given with DPT and OPV, the 3 doses, just like here, 6 week, 10 week, and 14 week. And it is really, really very important to remember this immunization schedule, not just as a doctor but also as a student, because it is very often asked in examination. So this is immunization schedule for national immunization schedule. Another immunization schedule is WHO EPI immunization schedule. It contains only 6 diseases: tuberculosis, polio, and diphtheria, pertussis, tetanus, and measles, in which we give at birth BCG and oral polio. At 6 week, we give DPT and oral polio. At 10th week, we repeat it, DPT, OPV. And 14th week, again DPT, OPV, and 9 months measles. In a way, it is similar to this part of our national immunization schedule. So this part of our national immunization schedule is just like EPI. Now coming to passive immunization. Passive immunization, sometimes because when we give vaccination, it takes time to develop immunity for that person, and sometimes we do not have that much time when a person is already suffering from a disease, or he is not suffering from the disease but he is a very high risk, or he has a very big exposure to a disease. Then we give passive immunization as a part of the treatment, and sometimes as a part of prophylaxis, when the exposure is very high. So in passive immunization, we give preformed immunoglobulins. In There are 2 types of passive immunoglobulins. One is homologous, that are produced in humans. The others are heterologous, which are produced in animals. The human homologous immunoglobulins, they have less adverse reaction, and their protection lasts for 3 to 6 months. The animal origin immunoglobulins or heterologous immunoglobulins, they give protection for few weeks, and besides they also have a risk of developing adverse reaction like serum sickness. Passive immunization, it can be of 3 types: normal human immunoglobulins, specific immunoglobulins, or antisera or antitoxins. Normal human immunoglobulins, sometimes when a person is highly susceptible for certain disease, what we give, we non-specifically we give normal immunoglobulins, human immunoglobulins, and they have some protective effect against measles and against hepatitis A. For example, if a person is traveling to an area where hepatitis A is endemic, so we can give them normal human immunoglobulins. But one thing should be remembered, that if you have given normal human immunoglobulins, any live vaccine should not be administered for at least 12 weeks. Second type of passive immunization is specific human immunoglobulins, in which these are made from the plasma of the recovered patient or immunized patient. Plasma from recovered patient or immunized patient, and they have a very high antibody content, and they provide immediate protection. For example, chickenpox prophylaxis, passive immunization against tetanus immunoglobulins, or hepatitis B, rabies, or varicella zoster virus. For tetanus, we use it for the treatment. We also use it for the treatment of tetanus, and we also use it for prophylaxis. Suppose there is a person who has a very a large dirty wound and there is high risk of developing tetanus. So what we do, we combine it with the active immunization. Antisera, when we develop antibodies or immunoglobulins in an animal, we call it antiserum. Antiserum. They are prepared in animals such as horses, rabbits, etc. When we need it in large quantity, we produce them in horses. Antisera are available against tetanus, diphtheria, botulism, gas gangrene, and also snake bite. Snake bite. Because in snake bite also the main mechanism of the pathogenesis is the toxin. The toxin. These antisera they occasionally uh, give rise to serum sickness. Serum sickness, it is a disease. It is a hypersensitivity disease, type 3 hypersensitivity, and in which a patient develop fever and malaise, etc. after taking a antisera. Because this antisera is from animal origin, so there will be certain proteins against which the human normal immune response will act and produce the disease called serum sickness. Combined passive and active immunization is given in tetanus, diphtheria, and rabies and hepatitis B also as a prophylaxis. So it is often given when we combine passive immunization and active immunization. It provides immediate benefit to the patient. As I said, if you give only active immunization, for example, there is a patient who has suffered a wound and you are suspecting that he can develop tetanus. So only giving active immunization against tetanus will not help because the patient may develop tetanus even before developing the antibodies against the tetanus. That's all for today. Are there some questions regarding this? So we talked about different type of vaccines, live, attenuated vaccine, killed vaccine, and subunit vaccine, RNA vaccine, recombinant vaccine, or we also call them a vector vaccine or toxoids. So advantage of live vaccine is that they give good immune response and a single dose is sufficient